Hello, everybody. My name's Mark Scheidler. I'm a chair of the CMTA Advisory Board, one of the advisory boards. And today I want to tell you in a short half hour a little bit about Alliance Initiative the CMTA has been working on for a few years now. And today I'm going to do it by giving you a brief introduction to the overall idea and then introduce a panel that I brought of people who are involved in this work and taking advantage of the capabilities we presented. And let me see, share screen. Okay, I'm trying to screen share. Okay, I think everybody should be able to see that now. So what I will start with is just introducing who's on the panel today. So I brought together Jae Young Lee, who's with Toolgen in South Korea. I brought together Michael Alajanian, who's with Orthogonal Neuroscience in Boston. And Toolgen's working in a novel CMT1A gene editing approach to treat CMT1A. And Orthogonal is working on a really interesting antibody approach for several different CMTs. In addition to that, Talene Hanani is here. She's really the head spearing the effort at a service organization called Psychogenics. It's doing a lot of the testing for us for these different therapy development studies. Oops. So let me just give you a little background on the Alliance Initiative as it is. So the reasoning behind developing alliances with pharmaceutical and biotech companies is that the CMTA recognized that the testing of a potential new therapy in animal and cell models of CMT was not generally or easily available. And to solve for this, we created toolboxes of these animal and cell models representing different CMTs and put them into use by a network of expert service companies who are very versed in how to do drug and therapy development studies at the preclinical or animal testing level. And the whole idea here is that by providing easy capability uh, to this capacity, and also the advisory know-how of the CMTA advisory boards, we could actually lower the access barrier to companies to the testing of new therapies. So if they had a therapy that they were trying to develop for one neurological disease and they had an interest in seeing what it would do in CMT patients, they wouldn't have to spend years developing this kind of capability to do it themselves. They could come to us and we could work with them to quickly get their therapy into an evaluation in a very well evaluated animal model. And the whole idea, again, is that companies that have therapy candidates that may benefit CMT patients can now use this capability to gain some enabling information that would really accelerate and promote the use of their therapy for the treatment of CMT. And this is a snapshot of our current service partners. And Talene's here representing one of the most key partner of Psychogenics, who's now done more than 100 studies with us to characterize the animal models and to evaluate therapeutics. We work and Psychogenics works in concert with the Cleveland Clinic that provides post-study analysis of nerve tissues to show whether a nerve is positively affected by a therapy. And there are a number of other important players involved as well. And the whole idea here that we're trying to reduce to practice, have been trying to reduce to practice, is that the CMTA as a disease, as a convener around CMT diseases, can pull together universities to provide know-how and also the models we need to use in testing. We can pull together contract service organizations and work with them to actually set up the testing in their organizations. And with those three things, the CMTA on the lead universities with know-how and the service organizations put together a network. Therapy companies can now enter into a consortium relationship with us in order to test a therapy to get it into animal testing. So let me see. So with that said, now I'm back on the screen, toggling between my own screen and this one. I want to just launch into a Q&A with the panel here and let you know what they have to say about working with the CMTA. So Michael, could you start out by telling us about the approach that Orthogonal is taking to develop therapeutics, and also about Orthogonal?
Yeah, thanks, Mark, and what a pleasure it is to be here today. So thank you for the invitation to participate in the panel. It's really pretty exciting for me and for Orthogonal. Orthogonal is a biotech company, and we're venture capital funded. So our venture capital partner is Morningside Ventures, which is in Cambridge, in Harvard Square. And we're a 5-person company, and we're virtual, which means we have no building, we have no brick and mortar. All of the work that we do is external with contract research organizations and contract medical research organizations. And so the start of the company was done by licensing in some technology from the Hong Kong University of Science and Technology, and then trying to make that technology, the research, practical and develop it into a therapeutic. And this is what we've done. The target for our company, the single project, is the ephrin A4 receptor, which sits at the synaptic terminals of the neuromuscular junction. So the neuromuscular junction is the end of those long neurons that come from the spinal cord and innervate muscle. And it is those neurons that tend to die back in CMT and other diseases. And so what the ephrin receptor does is actually exacerbate or make worse the dying back of those axons. And the antibody that we've developed, the therapeutic antibody that we've developed, actually blocks that receptor and helps the axon to recover and repair, and also stimulates neighboring axons to innervate the muscles that are undergoing atrophy or decreased innervation from the spinal cord. So what the CMTA has done for us, to be honest, is really helped open our eyes as to what the potential application of this therapeutic approach could be in CMT. And Mark was very helpful here, as was Talene. And they helped us to understand the animal models, which animal models we should test in. They helped us to understand what are the most relevant endpoints in those models, and they helped to design experiments for us. And so in collaboration with Mark and the CMTA and Talene at Psychogenics, we tested the molecule that we're developing now in 2 different models of CMT, one a demyelinating and one an axonopathy model. And the molecule had produced benefit in both of those models, not in every endpoint and not necessarily in the same endpoints in both models, but across both models, we saw a definite benefit produced by the molecule. And I just want to let you know that, based on these data and some other data, we're now in normal healthy volunteers in the clinical study, and that study should end probably in the spring. And if it turns out okay, then we'll probably look to start clinical treatment studies with our F8-4 receptor inhibiting antibody. I think that should be pretty much a nice background. Is that sufficient, Mark? Or?
Well, thank you very much, Michael. Jay, same question. Tell us about Toolgen and the approach you're taking to develop a new therapeutic here.
Yes, so thank you, Mark. So Toolgen, at Toolgen, we are trying to develop gene editing-based therapy, which means we are trying to edit the root cause of the genetic disease, which would be a PMP22 in a case of type 1A of the CMT. And thanks to discovery of CRISPR-Cas9, we can now pretty much edit gene of interest quite easily. And with this tool in hand, we are now trying to normalize the PMP22 gene expression for CMT1A by specifically editing genetic switch for PMP22 expression. So we are now at the preclinical stage, which means that we are trying to test our therapeutic candidate with in various CMT1A animal models. So just a little more background of the tools, and we've been around for just about 20 years. So we've been developing various generations of the gene editing tool, right from the zinc finger nucleases to TALEN, and now CRISPR-Cas9. So really our focus of our company is that we would like to translate this novel genetic technology or gene editing tool to novel therapeutics where needed.
Okay, thanks, Jay. Talene, tell us about psychogenics and how you're supporting these studies.
Thanks, Mark. So Psychogenics is CRO, and we've been established in '99, so we're 23 years old. Initially we started focusing on psychiatric disorder, but we've expanded quite a bit, and we've been working quite a bit with disease models. And we have actually done a lot of collaborations with disease foundations, like the Huntington Foundation, Michael J. Fox for Parkinson's, ALSA for ALS. So we've really had the infrastructure to work with the CMT, and we have developed a lot of already animal tests and assays to support some of the motor disorders and other functional disorders. So working with CMT actually has been really, really very interesting, and you have phenotyped several animal models from 1A to 2A, 2E, etc. And we've been working for years now, and as Mark said, we completed over 100 studies. Some of them are phenotyping new models, some of them assessing novel therapies, as Mark and Jay were talking about. And it's always fun because we always add more assays and we establish new models just for the CMT, and and it's been a very exciting project.
Thanks. You've also been a great partner in terms of pointing different companies towards us, because you have a big psychogenics is an excellent reputation in neurological testing, and people do come to you looking for how they can get this sort of work done. So it's been excellent in that regard. So, Michael, how did orthogonal become interested in CMT as a target indication for drug development?
Well, because we had this interesting mechanism of the compound, which seemed to help the neuromuscular junction repair and regenerate itself, we began looking at different diseases that could benefit from the modulation of the ephrin A4 receptor. And as we were doing our reading and our research, CMT came to the front, one of the front positions in that research. And so, based on the internal research that we did, based a little bit with discussions in discussions with Talene, and ultimately with Mark, we decided to invest some resources in testing the molecule in CMTA. But mostly, it was dependent on the mechanism of action of our target, and we felt that CMT patients could really benefit across all of the genetic variations of CMT, that they could all benefit from an ephrin A4 inhibitor.
Thanks. And Jay, I know Toolgen's involved in a portfolio of projects in gene editing in different fields outside of neuroscience as well. What put CMT on your roadmap? Why did you get into CMT as an opportunity?
Yeah, so It started from my personal interest, I would say. So from my doctoral study, I was heavily involved in discovering different mechanisms of demyelination in the central nervous system at that stage. So I was very much heavily interested in central nervous system diseases such as multiple sclerosis, which affects myelin and the brain and spinal cord. And when I moved to Tujen, I learned about a lot of aspects of the capability of gene editing technology, and I quickly thought that I can probably apply this technology to fix inherited myelin disorders with clear genetic cause. So from then, myself and team at Tujen, trying to develop some strategy for CMT1A, and luckily it worked in several model, and now we are trying to really translate this finding to the clinic.
Okay, thanks. And Talene, how does CMT fit side by side with the other kinds of indications you're testing at Psychogenics, and what specifically is the kind of testing that you do?
So absolutely. So CMT fits very nicely with many other disease areas we are working with, especially diseases that show motor function deficits, as well as muscle degeneration or nerve demyelination, etc. So we've been doing a lot of work with the CMT, looking in various endpoints. Actually, some of the behaviors we've been measuring have been focused on looking at fine motor disruptions, gross motor disruptions. We've been looking at gait changes. But probably one of the most interesting endpoint we've been looking at, and this is something we developed with, help with Mark, for the CMTA, is actually measuring muscle action potential and nerve conductance. So this is actually have been one of the probably more relevant endpoint that has better translation to human. There's always, some controversy going from an animal to human, and not all the behaviors obviously translate well. But I think looking at CMAP, looking at nerve conductance, I think these are more translation endpoint that we're looking at in conjunction with the behavior. So oftentimes we see some, in some cases, we do see some signals in the nerve conduction that we don't see in the behavior, or vice versa. So I think having this very nice sort of like a comprehensive approach, I think helped quite a bit with some of the drug screenings that we've been working with the CMTA.
Yeah, thanks. So as I see it, it's the usual suspects, in terms of translational clinical endpoints: gait, balance, grip, the things that really matter to CMT patients. And you can confirm what's going on with electrophysiology. And, there's a workflow here that moves on to confirming histologically at the Cleveland Clinic. And there are other partners involved in other parts of the work in terms of analysis, in terms of breeding animals. It is kind of a consortium effort between companies, small and large. So thank you for describing that a bit. So Jay, back to you a bit. Everybody in the CMT community is really eager for new treatments. Can you give us an idea of the sort of timelines you project that you're working with to get a therapy to patients, or just into clinical trials?
Yep, sure. So time spent on therapeutic development can be dependent on a lot of matters. So basically we need to go through heavy preclinical testing, both in vitro and in vivo. So in vitro means a cellular model, and in vivo means the animal model. And also we have to go through a characterization of our manufacturing. So it can be dependent on quite a lot of matters. But as CMT community is eager for new treatments, and so as us, so time is very goal for us as well. And at this stage, if everything goes well, we are trying to aim all the we are trying to gather all the data for clinical testing within a few years.
And Michael, you talked about having kind of 2023 as being a time where you'll be really looking at the ability to get into clinical testing once you actually with patients. Is that right?
Yeah, that's our hope right now, Mark. We should be finished with the normal healthy volunteer study, sometime towards the beginning or middle of next year, and then we'd like to be able to start a patient study sometime before the end of the calendar year next year. But I have to, stress to people that, especially patients, that right now we don't have the capital, we don't have the money to run a patient study. So part of the job of somebody like me and my boss, who's the CEO of the company, is to go out to venture capitalists and other people with substantial amounts of capital and try to raise money for these kinds of studies. And so our ability to run a clinical trial is going to be almost entirely contingent on our ability to raise money from potentially interested venture capital partners.
And just following on from that, with the therapy that you're developing, what benefit might somebody with CMT expect to see?
So the things that we've seen in the clinic are not in the clinic, in the preclinical studies are a couple of which were mentioned by Talene, the improvement in the electrophysiological measures like compound muscle action potential and conduction velocity of the motor neurons. But perhaps more relevant to the patients are things like gait and grip strength. We'd like to be able to see improvements in those kinds of endpoints, as well as the electrophysiologic biomarkers in perhaps a 6-month study. And we feel that that's possible based on some of the data that we've seen preclinically.
But do you think that this is going to target the ability to keep patients from getting worse, or actually repairing something repairing loss of function that's already occurred?
Right. So we believe, again, if the preclinical data are predictive, that both things could occur. So someone who's already been affected by the disease, who's experiencing symptoms of weakness or impaired gait, might be able to benefit within 6 months. And our hope is, based on the preclinical data, that improvement would be durable. In other words, with continued treatment, that improvement would be sustainable and constant throughout the subject's life.
Same question for you, Jay.
Yeah, so
I'm talking about, the kind of the target patient population. Are you looking for fixing damage that already occurred, or preventing it in the first place, or both?
yeah, so like I said, we are trying our best to basically fix the genetic problem. So although gene editing may sound ultimate solution for genetic disease, we really need to go through various preclinical and subsequently clinical studies to find out whether our therapeutic candidates can be effective in slowing down or halt or even restore various functions. So our goal at this stage is to at least provide a genetic handbrake, I'll say, to CMT1A progression. And of course, we will be testing if our therapeutic candidate can also provide a reverse gear to CMT1A progression.
Thank you. And back to you, Talene, for a minute. I think what we're describing here is the importance of time in developing therapies and advancing them to the patient. And money, of course, as Michael points out, is actually key to lubricating this going forward. And a lot of that's based on, can you de-risk this really early in the preclinical studies that we're doing? Because that really can accelerate an increase in investment interest, and also define ways to speed a clinical trial potentially. So, Helene, just give us an idea about the typical timelines and work path uh, that that Psychogenics uses in running a testing program for a chronic disease like this. And that's to distinguish this from other neuropathies that may be acute chemotherapy induced. But this is really a chronic treatment that we're trying to develop.
Yeah, I mean, so these studies are, long-term studies, they're longitudinal studies, but, with the CMTA, we already have, colonies, we maintained, breeding colonies of the various models that we are using, for the preclinical testing. So we trigger a study, we trigger a bigger breeding once, we have a study. And typically, depending on the model, we can start the treatment as early as when the animals are 6 weeks of age, and it goes all the way, until the animals are, 21, 22 weeks of age in some cases, or in the case of CMT2A, that can go obviously longer because, it starts later. But these are these are about, like, each studies, they're about 12-week studies, sometimes 18-week studies. And along the way, we look at, half points as we get some data to see where we are, how things are going. So these are long studies, but that's how you have to see the disease progression, and to basically see if your compound is delaying, the progression, or if it's just reversing some of the symptoms. So that's why, these are long studies and need to be planned, very well.
Yeah, so I could just chip in here that I think that trying to get outcome measures on a chronic therapy in 6 months is pretty fast. And that's something we've worked to. And what's helped us with that is doing the kind of profiling of the animal models over time to see what's affected with age and when. And that really informs the study design. It allows us to do a study that for chronic diseases, a very reasonable timeline. The other thing that you mentioned is actually having teed up groups of animals in breeding that allow us in one generation to get into a pretty big study has been strategically, I think, very important to make all the wheels move here.
Yep.
And I can say that on the post studies that we do with Cleveland Clinic, there's a similar emphasis on timelines in terms of stages of what's the most important data, and let's try to get it to you as fast as we can. And they've developed some novel algorithms in their imaging systems to help accelerate the analysis of the samples. So that all taken together, let's go back to the clinical side for a moment. Just trying to paint a picture of kind of how preclinical testing can aid in de-risking a therapy to help it get into the clinic and hit those key influencers involved in investment, and also in determining whether something's likely to be feasible to actually treat a CMT patient. So Michael, what group of people do you think could be treated with therapy? Young, old, male, female, early, advanced symptoms? Would they be treated short term, or need to continue with treatment over time? So, or is there the possibility of, something in the middle, that involves drug holidays? Just kind of paint a picture about an antibody therapy and how it looks.
Right. So we believe that based on the mechanism, we ought to be able to treat most subjects, whether it's early or late in the disease, because we feel that the preclinical data have shown us that we can actually get improvement in some of these outcome measures like grip strength or like balance, rather than just slowing the rate of progression of the disease. So based on those kinds of data, we think that we can treat most subjects. Now, that said, if a subject has severe disease, where the motor neurons coming from the spinal cord have significant amounts of loss, then our mechanism may not be helpful there because they actually need intact neurons. The mechanism requires intact neurons to have a positive effect. So, while the most severe patients may not be included in a clinical study that we might execute, certainly the mild and moderate patients that have intact neuromuscular junctions, that have, intact motor neurons that interact with muscles, we think that they could be potential candidates for any clinical study. And, for better or for worse, we believe that this would have to be a chronic treatment. And what that would mean for an antibody is an IV infusion, probably once a month or once every 2 months. So perhaps a nurse comes to your home and gives you an infusion, or you go to a local clinic and get an infusion. Those are the kinds of ways that we predict that this treatment could be administered to patients in a chronic way.
Thank you. I'm going to turn to Jay, and what I'm really asking is, what does your target product profile look like? Which is kind of clinical roadmap a company builds around when they anticipate getting into a new therapy area.
Yeah. So our therapeutic candidate is not designed to be limited to certain populations. So basically, when we're trying
For
to
CMT1A, excuse me, for CMT1A. So
yes. So for
right.
CMT1A, it really is if any subject has diagnosed with PMP22 duplication in the genetic information, they will be pretty much eligible. So, however, we will need a lot of data readout from the animal model studies, and of course to consult with various key opinion leaders in the field to decide which population could be first be treated. And I heard from the previous panel discussion at the gene therapy session that adult population would be the first participants to be trialed, simply because the young patients would be there will be a little bit of complication between developmental aspects and the pathological aspects. And one key advantage of in terms of the how many doses that need to be treated. So one key advantage of gene editing based therapy is that you only need one-time treatment for durable therapeutic effects. Having said that, we do need further data to support durability of therapeutic efficacy, but theoretically, therapeutic candidate only need to be administered once.
Okay, thanks, Jay. We're going to have to wrap up here. I'll just make one final comment, which is what we've really been describing here is a staircase. How do you walk up the staircase from a basic research discovery into testing in animals of a potential therapy based on that discovery that validates that this is a good approach, and into clinical studies. So Jay's kind of finished up talking about next steps will be recruiting a clinical advisory board around the project to help them develop the therapy. And that's really what the CMTA is trying to offer, is these different steps in the staircase. And with that, I'm going to leave it off, and thank you all for joining us today.
Thanks, Mark.
Great. Thank you, Mark. And thanks, everyone.
Bye.